So today, well, I'm talking about the vaccine research, violin research. I'm more actually proposing a new common data model for things beyond, can be used beyond vaccine. I want to emphasize this point because then everyone can be potentially useful. So also another difference is like we have been focusing on ontologies. Ontologies are more for knowledge collection and representation, right? So here actually we are more focused on experimental data and how we can integrate the experimental data by using something called a common data model. So the good thing is like we have many talks, right, ahead of me who have giving wonderful information and give enough background for all of us about ontology, standards, knowledge graph. So I don't have to introduce them. Again, we have many resources, right, in here, like something related to vaccine or immunology. Then we have import, right? We have other more general ones like cell by gene. Like I also been working with the kidney precision medicine project and HarvardMed and our violin vaccine resource. So if you look at them, each of the resources have generated a lot of data, right? So we often think about how to get knowledge, but then the fundamental question is like how to represent the data. So one good example is the OMOP. So OMOP has been very, very popular over the last about 20 years. So like, for example, I'm also using the N3C data. It's just one single data resource is managed by NCATS, right? So it has over 22 millions of records and billions of rows and data, right? So, and they coming from like about 100 hospitals. So you would be wondering how good, how can they do, right? So they are handling EHR data. So it's not knowledge, it's more like a data. But then the EHR is EHR, right? What about our biological or biomedical experimental data? So they don't handle it. They don't target handle it. So then the question for us is like, can we generate something like OMOP CDM, but not for EHR, or can be for EHR, but most importantly, can be for the experimental data. So I look around, right? So there's something called the ESA template. So ESA is investigation study essay. So people use it for a while, but things like it's a little bit coming down to not so many people use it anymore. So, and we are kind of feel like, okay, what is the difference between investigation and study? Right? We get a little bit confused. And it's also a kind of semantic model. We try to be more like a OMOP, right? It's more like a relation database model or knowledge graph model. So we think we can reference them. We probably don't use them. And then there's an import data model, right? Import, like if you are working on immunology, right? You probably have used it. It's very popular. So import has more than 33 tables. So it's very good actually for immunology, but maybe it's not good for some other domains beyond immunology. So how can we learn from it and generate a more kind of generalizable model, right? So if you look inside the import data model, you find actually something you can kind of put together. So for example, they have essays, right? They have 15 essay tables. So you feel like, oh, they're all essays actually. You can just use one table called essay. So you can include them all. Another example is the interventions, right? They have 3 tables. So one is called intervention, one is called immune exposure, like a vaccination, right? Immune exposure, or treatment, right? Like a drug treatment or some chemical treatment. So you can actually think, yeah, maybe we can simplify that. Let's just call it intervention, right? So they are all kind of different kind of intervention. So when you do so, actually you can use it for many other interventions. So it's like how to make it more like a first principle, right? To simplify the story. And then also samples, right? So in the import, they have 3 types of samples, like experimental sample, and clinical biosample, and then control sample. But for us, we feel like, oh, they're all samples, right? So you can classify them as a sample. So you see, I'm going to, right, to make it simple. So then the first general idea is like, let's call it SEA, right? So it's a study, experiment, essay. So we call it C, right? SEA, C common data model. This is more like a basic idea. Later on, I'm going to give a little bit more kind of a comprehensive idea. So the basic idea is like they're more like 3 processes. So you have studies overall thing, right? So one study can include 1 or more experiments, and then experiments experiment involved in organisms like human or mice, and then vaccination or other intervention, right? And then in the end, you have samples. Sample is the input of assays, right? So this is the difference between experiment and the sample and assay. And then all these can have different experimental factors. So those factors can indeed likely affect the results. So you have to record those factors in. So that is more like a basic idea. So it was presented by my PhD student last year in a workshop. And then we kind of feel like, yeah, it's not that mature. We further developed it, and now I'm presenting an updated version. So let's go further on the C model, right? Again, study, experiment, assay, and then organism, intervention, and sample. And then we put a few more tables, like a group, right? So you can group, for example, you have a group of mice, like a vaccination group and then control group, right? And then you can use it to collect the groups, and then you can do some statistical analysis, and we can put some statistical analysis methods and factors, right, about it. And then in the end, you have results. And then we have material like vaccine or drug or chemicals, right? And ontology, I'm going to mention a little bit more, and then documentation to give protocols, those things. And then, so now that's the table level, and then you go to the attribute, the column level, right? So we just find some examples like organism. You have like age, sex, species, race, etc., right? And then if you are using mice, you have a string, right? And then in intervention, as I said, right? Intervention can be like intervention time, like day 1, day 7, and then dose and the route, and those kind of intervention type. Then you say, yeah, you can use intervention type, a magic type, right, to cover, say, is what kind of intervention it is, right? And then you have samples, when to collect samples, and assay and the results. So, and then you can use IDs to link each other, link different tables. And then I think another pillar of the whole system is ontology. Again, I learned from OMOP. Indeed, OMOP CDM use ontology a lot. I'm indeed a co-chair of the vaccine vocabulary working group in OMOP. So I have been working with OMOP for years. So I kind of know how ontology can be important. So in our CDM model, so we envisioned it's a little bit different from OMOP, because OMOP is self-contained. All the terms inside is a self-contained system. We feel like maybe we can be a little bit kind of flexible, because biomedical studies, they are all kind of areas, right? So we can use, like, for example, my lab has been developing the Ontology-Based program for years. Actually, a lot of people are using it. We have a Sparkle query. It's a very nice feature. So, we can use it, rather than have its own self-contained, so make it kind of loose. So just give one example. So my lab developed the so-called vaccine ontology. We now have covered more than 10,000 vaccines. So all like human licensed vaccines, all licensed animal veterinary vaccines, and research vaccines, and vaccines at a clinical trial. So we can use that one. This is just one example to show how the trivalent influenza vaccine. I'm going to use it as an example later, so you can see there are a lot of the other terms. There are specific trivalent influenza vaccines. And then you can do a Sparkle query, right? So you just use Ontology-Based Sparkle query interface. A few lines. So you find, 771 vaccines under the trivalent influenza vaccine category. That's kind of amazing. So you can use it as an example. So you can use ontology to organize data. And then I'm going to try, in addition to the common data model, right? As I said, we're trying to more like follow OMOP idea. So we build up a recent database, and we use the VGET as a first example. So VGET is all like a vaccine information. It's more instance data. We put it into our Violin resource is a part of the violin program. So it has a lot of data and a lot of genes plan data and a lot of studies. So we're trying to say, okay, let's make it as our first use case. So actually, like this is also our own program. I was working with Dr. Guanmin Wu from Oregon. So let's use this one as an example, right? So let's use the CDM model. So this is exactly the same idea of the CDM model, but kind of for each study, experiment, assay. So you have specific values. So we can put them into a database. And again, this one is a fun part. So my student, like Anthony, so he gave a name like there's a typo. Pelagic. Yeah, pelagic is more like the open environment of the sea. So basically, it's a Python program. So it actually kind of query we call it ocean, right? Ocean is ontology-based C, SEA network database. So we put it in the database. It's more like a metadata, but the raw data, the genes plan data, it's more like an individually file. But the database records the information about the raw data, the normalized data, indeed. So you can have a program, right? You just write a simple in your Python, you embed some SQL query, SQL query, right? So you basically say, okay, I have an example. What are the genes stimulated by fluorix influenza vaccine in females at day 7 after vaccination? Just one simple example. So I can write a few lines of code actually, and the first part is more like the metadata, and the second part is more like how to define the restriction of the genes, right? So you can get the gene data, you can pass it. So you basically just a very simple example, very simple story. Then you can actually identify the list of genes with the average gene expression retrieved. We have like a CSV file or JSON format, so you can get data. I'm going to give you some example results next. So for example, in here we look at like the influenza example. So we found a lot of vaccines, right? If you use vaccine ontology, we know those vaccines are inactivated or killed influenza vaccine. And there's one vaccine is a live attenuated vaccine, right? So you can actually identify all the genes like in female or male. And then we can look for the GSEA. We can actually indeed find a lot of patterns. They make a scientific sense. You can find like the live attenuated one indeed is quite different from the killed vaccines. And then we say, okay, let's go from the weaker model. Let's use more data sets. So we use import data and we use cell by gene data. And we put all the import data into our system. And we give some like competence questions. We say how many influenza infection, right? It's not vaccination, it's infection samples from these two data sources. So you can get all the data there. They're quite different, right? And then in addition to the MySQL region database, we also generated a knowledge graph. So again, it was introduced earlier, background. So you can see using the trivalent flu vaccine, right? So in here, actually, because the knowledge graph includes the vaccine ontology, so you can automatically, using a cipher, you can query the vaccine. You find a lot of vaccines. They don't have the name, but they are indeed a subclass of the vaccine type. So and then we can use the Neo4j. You can explore, you can identify the metadata. I think it's cool. But anyway, together, so we developed a CDM. We think it is very good domain neutral CDM. Also, it has been tested in vaccine and infection. We developed an ocean database and also a knowledge graph format. And we developed some pelagic Python program, so we can actually easily integrate the query and analyze data. We have 3 use cases. So we think they can be used for other domains. So we're doing testing. I think it's a very good one because theoretically, those levels are very high. So you can use them for all others. Okay, so I want to thank you. Thank the U24. We are currently developing. We are currently being funded by Ishiva's program. So yeah, we are working on the project. We have a lot of collaborators here. I mentioned, yeah. Just before I end, I have an advertisement. There's a meeting called the International Conference on Biological and Biomedical Ontology. It's all virtual, and so you can actually say if you are interested, you can submit papers or abstracts. Yeah, thank you.
Any questions? The only thing that Holding is back from lunch, I guess.
Yeah.
He'll be around the rest of the day if you have other questions.